Start with redox. We got people waiting. Now you guys are experts on redox reactions? Great. That's awesome. Okay, so remember what we're going to see several times in glycolysis are redox. Redox reactions are all about just moving electrons, which are energy, right? So we're going to start with glucose, and we're going to do this kind of step-by-step process where we start to break the bonds, release the energy. We lose some of it, right, at every step. But the cell is going to try to capture that energy in these redox reactions so we can do work with that energy. So we want to try to keep in mind what's going on with these electrons, how they're being transferred from compound to compound, and how NADH, remember we talked about, that's kind of like the taxi cab. It's going to collect this energy, and then later on we're going to see where it delivers it to. We won't see the whole story today, but yeah. Okay, that's the rule. Just remember, we'll see NAD plus going to NADH, so we'll review it as we go. And this is kind of what I was talking about. Instead of having a huge release of energy all at once, and you can capture very little of that, the cell is going to do it in a stepwise fashion. So glycolysis is 10 different steps. Many of them are redox reactions where they're just slowly extracting this energy. Okay? The other redox reaction, so this is one you should know. So NAD plus can be reduced to NADH, and it can go back again as well. This is one example. The other redox reaction, technically, we're going to see is this reaction with ATP. So we're going to use ATP as a source of energy in one case. And the way you do that is you can actually chop off these phosphate bonds. There's 3 phosphate functional groups on each of the ATP molecules. And that represents high energy electrons. There's a lot of energy in those bonds. So what we can do, not we, but enzymes, can cut those phosphates off. Usually it's just the first one, but you can cut the other ones off as well. But you can cut the phosphate off, so you go from ATP, which is triphosphate, to ADP, which is diphosphate, so 2 left. You cut it off and you can use that as energy. Cutting that bond is energy. That functional group can be energy. We've seen this before. Active transport. Which one, what protein was it? Yeah, sodium potassium pump. Chopped off a phosphate and stuck it to itself and used it as energy to change shape. So it can move things in opposite directions, right? Okay, so that phosphate group is energy. It can be used as energy. But there's a cycle. So you can use ATP for energy to do work by chopping off that phosphate. But then you can re-energize it as well. So in glycolysis, which is a catabolic reaction overall, it's a catabolic pathway, we're going to see that we're going to take ADP, attach a phosphate to it, and re-energize it back to ATP. So this is technically another redox reaction that we're going to see. We're capturing electrons, using it to form a bond between the phosphate and the sugars. We'll see this in a second. So we're going to use that phosphate as energy, but we can also put it back on ADP to make ATP. Okay, so I just group them as redox reactions because we're just moving electrons. What we're doing, one way or the other.
How does the cell make it like produce more energy than is required to add the phosphate onto the ADP?
Well, so he's asking how is it possible? By breaking this bond here, this energy released gives you enough energy to add that phosphate to another compound as well as produce heat. So they're coupled. You have to produce enough energy in order for the enzyme to do whatever it's supposed to do.
Right.
It usually is more, provides more.
It doesn't take the same amount of energy to add the phosphate back on the ADP to make another ATP?
Oh, does it use the same energy? No. This will actually be lower. If you look at the kilojoules, yeah. And that's because they're inefficient. We're losing heat as part of this. So you have to have more provided in one direction. But we won't look at the actual energetic. You can do that in biochem or something. Actually look at the numbers. Okay, let's dive into it. You ready? Woohoo! Yeah? Maybe? Oh, by the way, yeah, glycolysis is only part of metabolism. So try to remember these pathways are not working in a vacuum. These things are not working, and they're not lined up in a nice long line either, right? These things are floating around randomly in the cytoplasm or wherever they are, because it could be the mitochondria. But they're floating around, bumping into the substrates randomly. So we're only looking at little pockets of this, but this is where you have to start. You look at the pathway by itself, and then you consider what it interacts with. It breaks it down into pieces. Okay, glycolysis is the first of 4 pathways that we're going to look at in aerobic respiration. Today, we're only going to look at glycolysis, which occurs in the cytoplasm. And we're going to look at fermentation, which is actually an alternative to this pathway, because there's going to be a decision point after glycolysis. But we're going to look at each of these. You need to memorize their names. As we go through them, I'll tell you what things I want you to memorize from each pathway, okay? So that gives us a kind of a foundation as we're talking about the overall metabolism of the cell. The other thing to realize is the big goal here, make lots and lots and lots of ATP. That's the overall goal. Because ATP is going to be our energy currency in the cell. What most proteins are going to use for energy to do whatever they're doing. Okay, glycolysis. It's actually split into 2 phases, or 2 halves. So I'm going to draw this on the board, and we'll just kind of go back and forth between this picture and the board, okay? So there's an energy investment phase and an energy payoff phase. So this is glycolysis. Remember, it's all about breaking down sugars. We start with glucose. That's the preferred energy source, not energy source, preferred sugar source for most organisms is glucose that starts this pathway, which we find in most organisms. I'm just going to use several arrows here because we're not going to look at detail at every step. Okay, so we're going to kind of make it a little easier. We're going to take glucose, cut it down, and halfway through we come to a compound called 2G3P. And this is what I consider as kind of the halfway point of this reaction, is the G3P. And then we're going to take that further through several different steps and produce pyruvates, 2 pyruvates. So this is kind of what happens with the sugars. And then on this half, we're going to see this is the energy investment phase. And then the second half is the energy payoff phase. You guys have heard the term, invest a little, gain a lot? Huh?
Which that board at?
I didn't hear you.
Nothing.
Was it a joke? Yeah. And then we lost it because I couldn't hear it? Okay. I'm sorry. I'm sure it was an excellent joke. But it's like investing, right? You earn a little bit of money that you expect to have a big payoff again, hopefully. You guys actually should be investing already, right? Because Social Security is probably going to be gone by the time I retire. It's going to be way before you guys. Okay, so you should be investing now. That's why you're in college, right? Did a good job, steady career, lots of money. Fame, fortune? No, I'm so yeah. So, same thing with the cell. The cell is never going to make any kind of investment or burn ATP for no reason. For no reason at all. Oh, thank you. Very distracting. Okay. The cell's never going to waste any energy for no reason. It's always going to use that energy for a reason. But we're going to see here in the investment phase, it actually burns a few ATPs to start the process up. It's almost like that idea of priming a pump again. We're going to put a little bit of energy into the process, but we're going to see it's going to pay off. The form of the energy that it puts in is ATP. So we're actually going to see we use 2 ATP in this first phase. We're not looking at individual reactions here. That's going to make 2 ADP. So we're going to see that we use 2 ATP. So notice, when you go from ATP to ADP, you're using energy. You're investing energy, okay? You're breaking that phosphate bond. We're going to invest a little bit. And then in the second half, we're going to have a payoff. So the second half, we're going to see that we make 2 NADH. So we go from 2 NAD plus to 2 NADH. I'm just going to put these in boxes. And then we also make ATP. So we actually make 4. So 4 ADP going to 4 ATP. That's going to be our payoff. So investment, we use a glucose, we put on a couple of phosphates. We actually end up with 2 sugars at this point. And then as they continue individually through the second phase, we start getting the payoff by making these energy-rich molecules. So we're making NADH and ATP. This is the kind of the basics of what I want you to know. So tonight, when you go to practice in the library, you draw this picture with a bunch of empty squares or boxes, and you start filling them in. This might show up in the quiz next week, okay? So you want to start memorizing them, certainly for the exam in a couple of weeks. You want to know all these and the numbers with them, okay? I know it sounds like a lot, but it's only the beginning if you're going to continue on into biochem and all that stuff, okay? Let's look at the individual steps. So this is what I drew, okay? It just has a little extra information on it. You have the ATP used at the top. You guys see that? ATP going to ADP, used to. And then here we go backwards, 4 ADP to 4 ATP, 2 NAD plus to 2 NADH. Yeah? So far, so good? Let's look at the individual steps then. We'll come back to that. Whoops, there we go. Here is the first half, the energy investment phase. So you see that the first step here with hexokinase, that's the first time you use ATP. And then the third step, we use another one. And you can physically see the phosphates being added to the sugar molecule. So the phosphates being taken from ATP and put on the sugars. So we're investing money in this to ATP. Part of the reason is we don't want the glucose to leave the cell. How did the glucose get into the cell, by the way? We talked about digestion a little bit. Goes to the bloodstream. How does it get into the cell? What kind of transport? Yeah, active or at least facilitated, doesn't it? Yes, because there's a whole bunch of hydroxyl groups on there, which makes it polar. So it will not diffuse through the membrane. But glucose comes in through a transporter. It can go back out of the transporter as well, unless you modify it. All you have to do is stick a phosphate group on there. That actually does not allow the glucose to go back out the transport protein, because it's not glucose anymore. Glucose 6-phosphate. Kind of interesting. So just a small modification will change the molecule enough. Watch out for those garbage cans. Just a small modification will change it enough that it won't be transported back out. Why? Does anybody have a problem with this energy investment? How did I describe glycolysis in our intro lecture? Overall, what is it supposed to be? Yeah, 10 steps. Well, is it catabolic or anabolic? Is it endergonic or exergonic? That's a trick, right? Because the whole pathway is supposed to be catabolic, but that doesn't mean every step has to be catabolic. Or exergonic, if you will. Okay? See, I dodged it that time. Okay, look at this energetics here. So this is G on one side, this is aerobic respiration. The beginning of glycolysis, what happens? What type of reactions are we dealing with energetically? Yeah, is it going to the plus or the minus G? And that means what? Endergonic. Endergonic. So by putting those ATPs or those phosphate groups on there, our energy actually goes up. I don't know, to plus 20, whatever it is. It goes up. Plus. But then what happens? By the end of glycolysis, where are we at? Minus 100. And by the end of aerobic respiration, where are we at? Can you see it? 600 minus 600 something? I don't know. That's an investment return I'll take. Yeah. Putting in 20 bucks and getting out 600? Yeah, good luck getting that nowadays in a savings account or anything. I think my savings account has 0.6% on it right now. It'll be that way for the next couple of years. So that's not the way to go. This is more like, what, roulette? Not Russian roulette. No. Something else. Okay, but do you see, are we getting a payoff though? At least we're not going to look at all this yet. But even at the end of glycolysis, we still have a minus 100. So that's pretty good. So again, the cell's not going to do that endogytic process for no reason. It's going to invest that little bit to start it, but then it's going to look for a big payoff from that, okay? Okay, so first off, we invest 2 ATP. So far, so good? And our halfway point, everything is being converted to this right here, glyceraldehyde 3-phosphate, or G3P. You actually get 2 of them. You get 2 of them. So we lost any carbons yet? That's a good question. How many carbons did we start with? Anybody know? Yeah, this is a 6-carbon compound. And if you look at glyceraldehyde 3-phosphate, it's 3 carbons. But we have 2 of them. Have we lost any carbons yet? No, 3 times 2 is 6. So haven't lost any carbons yet. What have we lost? From the sugar. Have we lost anything? We haven't even lost electrons, have we yet? Not yet. We've added phosphate groups to it, haven't we? Okay, good. So now we're going to see the payoff. How are we doing? Okay, payoff phase. So you have how many redox reactions here? Well, this is glyceraldehyde 3-phosphate going to bis-phosphoglycerate. So what's this? NAD plus NADH? That's redox. ADP to ATP? Redox. ADP to ATP, redox. So 3 different redox reactions. Here we're stripping off electrons like crazy from the sugars, okay? So the first step, this is right after the midway point. So right after G3P, this one right here, this is where you produce the NADH, okay? That's the first step. And then in 2 successive steps here and here, you're going from ADP to ATP. So you're taking the phosphate groups off of the sugar and putting them back on the ADP to re-energize that to ATP, okay? So this is our payoff. Again, you don't have to memorize the individual steps where they occur, but you want to look at the overall kind of net gain from what we get from glycolysis, okay? So what do we get? What is our net gain? I'm going to move my camera. What sugars are produced? At the end? 2 pyruvates. How many NADH do we get? 2. How many ATP do we get? Net. 4 minus 2. So 2. Okay, so now realize these ADPs, they don't have to come from this. Okay, you have to realize there's a lot of ADP and ATP floating around the cytoplasm, as well as substrates and enzymes and everything else, okay? So those don't have to be linked. But when we look at the net, we do want to subtract the ATP because we used 2 and we gained 4. That's it? 2 whopping ATP from a glucose? Yeah, so far. It's kind of wimpy, but this is energy, this is energy. We're going to see that NADH is going to be transported somewhere. It's going to take that electron somewhere. That's energy. And we're not done with the sugars. The sugars are energy too. If you look at pyruvate, which we'll look at the structure here in a sec, hell on, it still has lots of bonds on it, lots of high energy bonds. Questions on that? Okay, how about expectations for glycolysis? You have to memorize this, along with the numbers. And you have to have them going in the right direction. So when you go NAD plus to NADH, you have to have it in the right direction. It's not the same if it goes NADH to NAD plus. That's an opposite reaction. Okay? You gotta get the numbers down and all those what do you call it, cofactors and everything down. Oh, and by the way, we still haven't lost any carbons. There's still 6 carbons over here. We still haven't lost anything. At the end of this, we've only lost electrons. Or gained them on ATP and NADH. Okay? Yep, I know it's a lot. But like I said, put it on the board in the library and start filling it in when you're in your study groups. From memory. Okay? Yes, ma'am?
The middle, is the 2 to mean halfway, or are there 2 G3?
There's 2 of those molecules. So 2 molecules. We started with 1 glucose here, 1 6 carbon ring. Here, there's 2 molecules of a 3 carbon sugar. So if you look at the structures, which you don't really have to know the structures, but if you look at them, in glyceraldehyde 3-phosphate, there's 3 carbons. And in pyruvate, there's still 3 carbons. So it's kind of an accounting. You guys are an accountants here. Where you're trying to keep track of all values and all the sugars. Yeah? Okay, any questions on expectations? Again, when you hit biochem, they'll make you memorize every sugar and every enzyme. For some reason, I have no idea why. But they do. We all have to do it. I guess be happy for now, but you only have to do kind of the basics. Kind of. I don't know if that's any consolation. Sorry. But do this. Don't look at that. Do this right here, all right? So if you do that over and over and over again, you can fill that out from memory, then you're golden, yeah? And understand what's going on in each reaction, I guess. What's going on in the redox reactions. You guys good with that? Well, I don't know, that's the wrong way to say it. Is there anything else I can say that will clarify it better? How about that? Yeah.
Will you explain what redox is again?
Redox is a transfer of electrons. So if you go back and look at, if you look at this one, so like NAD plus can be reduced to NADH, and we'll see NADH can be oxidized back to NAD plus. So redox reactions involve moving electrons. And so the way to practice that would be to, you could even use one of these reactions in glycolysis. And you could say, what is it? G3P goes to bisphosphoglycerate in that reaction. So one way you could practice this would be to draw out the equation. So G3P goes to bisphosphoglycerate. So this is the middle reaction right after G3P. And what's the redox? This is a half reaction. So the sugars are being what compared to the cofactor? So you should be able to draw something like this and analyze it. What we did on Friday. So which ones are being oxidized, which ones are being reduced? Because they always go together, right? You have a reduction, you have an oxidation. Yeah, the sugar's being oxidized because the electrons are coming off of it, which means NAD plus is being reduced to NADH. So to get full credit, you would say 2 sentences or 1 long sentence. G3P is oxidized to form BPG, while NAD plus is being reduced to form NADH. That gives you full credit on an exam, because you described both of the half reactions. Does that help? So really all it is a transfer of electrons, isn't it? We're trying to follow the electron. That's what you do in OChem. You buy a book on following electrons. That's what you do in organic chemistry. So this is organic chemistry, really. So I would practice that. When we do citric acid cycle on Wednesday, you'll get more examples of redox reactions. So just draw them out and think about which way are the electrons going. Does that help at all? Or?
Yeah.
Okay. Maybe a good video. I don't know. You can probably find a good YouTube video or Khan Academy video on it as well. Okay, folks. A couple more things. So one, when we looked at the second part, the energy payoff, we see our first examples of making ATP. This has a fancy name. It's called substrate level phosphorylation. So it's where an enzyme takes a phosphate group off of a sugar. So phospholipyruvate is the second to last substrate. Takes that phosphate off of the sugar and puts it onto ADP to make ATP. So that's how we get our 4-ATP at the end. It's through substrate level phosphorylation. We'll see later on, this is covered up by the Zoom thing, but maybe on Wednesday we'll make it to oxidative phosphorylation. That's where we're going to make a bunch more ATP in the mitochondria. So there's a couple different ways we can make ATP. The first one is this here, okay? So just remember that fancy name. That's a way to make ATP from ADP, okay? So it's the terminology. Okay, now we're at a decision point. So just like our homework, we're going to encounter a fork in the road after the end of glycolysis. And it depends on how much oxygen you have in the cell. That's what regulates this pathway. I'm going to move my screen over. That's the wrong one. Sorry. So just like our homework, right? In the homework, we have forks in the pathways, which could be regulated by metabolites or whatever, inhibitors. Same thing here with glycolysis. So from glucose, now I'm just going to draw glycolysis as one big line. This is in the cytoplasm as well. So in most organisms, what we do, I should put the pyruvate, sorry. We have two pyruvates. What happens in these pyruvates is dependent on oxygen. So if we have oxygen or sufficient oxygen levels in the cell, we're going to continue with aerobic respiration. At least in most eukaryotes, we're going to go that direction. And that's because that's where we're going to get most of the ATP from our sugars. It's through aerobic respiration. We're going to get a lot of payoff. Okay? But there are some situations where we don't have oxygen. Sorry, I should put it next to it. Where oxygen levels are low. The alternative is to go through fermentation, which is a form of anaerobic respiration. If you go into micro, you'll see there's actually lots of different types of anaerobic respiration that bacteria can do. So they can make all sorts of products from that. We're going to look at a couple of different types, alcoholic fermentation and lactic acid fermentation. And this is interesting, though, because fermentation, the byproducts of this are actually lethal to the cell. So we have kind of a conundrum of why we would even want to do this, but I'm going to tell you why here in a second. But this regulatory point, by the way, is due to the transport protein that takes pyruvate into the mitochondria. What kind of polarity do we have on pyruvate? Polar and charged, yeah? So is something like that going to diffuse across the mitochondrial membrane, which is a plasma membrane? Or a lipid bilayer, how about that? The lipid bilayer. Yes, no, maybe? Polar, no. It has to be facilitated by a transport protein, okay? So interestingly enough, the transport protein on this is controlled by oxygen levels. How cool is that? Or it's at least sensitive to oxygen levels. So when oxygen levels are high enough, it will transport the pyruvate into the mitochondria, which we'll see on Wednesday. If not, what does it do? It shuts down, just like our pathway normally can. And what happens to pyruvate levels in the cytoplasm? This was on the homework, right? You shut down the two forks, but levels of O keep going up. And then it has a cascade effect. What happens to pyruvate levels? Somebody mumbled it. Yell it out like you love this stuff. Not really. It goes up, doesn't it? Because it has nowhere to go into the mitochondria. So that's the trigger for fermentation. That's the trigger for fermentation. So what are the fermentation pathways? So here is alcoholic fermentation in yeast. We go through glycolysis. And then the fermentation pathway converts pyruvate to ethanol. Okay? In humans, we take pyruvate and we produce lactate from that, or lactic acid. Okay? Interestingly, these end products are lethal. Excuse the story, but I always pick on marathon runners, so sorry if you're a long distance runner. But this happens when you run long distances or when you work out a lot. So you're, I don't know, let's say you're running. Part of your training is learning how to breathe properly, so you try to keep your oxygen levels high. But it can happen where your muscles start to get starved of oxygen, yeah? It can happen. So what happens to aerobic respiration then if you're starved with oxygen? Back to this one. This is aerobic. It shuts down. Okay, well, you need ATP in the cell, yes? You have to make ATP, otherwise that's going to be basically cell death at some point. So your brain is telling your muscles to keep going, but the muscles are being starved of oxygen. And they don't want to die, so what do? They switch to fermentation, lactic acid fermentation. And this is the question. Why? What does that provide the cell? It's kind of like a short-term alternative pathway to try to avoid death, basically. But what is it actually doing at the pathway level? Can you guys see in this picture? Well, now this is interesting because fermentation doesn't actually produce ATP, but it does something for glycolysis. Let's write it out on the board. Yes, that's a good key word, recycle. What is it recycling? Ah, yes, the NAD+. Okay. So let's write it like we had it on the board or on the picture. So I'm going to redraw it. So glucose goes through glycolysis to make pyruvates, is that right? And we get what in terms of ATP from that? 2 ATP, yep, something you want to start memorizing. And then we take the pyruvates through fermentation as humans, and we make lactate, which if it builds up enough will poison your cells. And that's kind of a sad part of marathoning, isn't it? A couple years ago in Boston Marathon, somebody died, didn't they? Why? What muscle do you not want to stop working? Yeah, your big heart. If it fills up with lactate, you're dead. If those muscle cells stop, okay? So this is weird. Why would we do this? Well, what was that part in the middle? Somebody said, recycling what? From glycolysis, we make 2-NADH, don't we? Which normally would be used in aerobic respiration. But this is not aerobic. Remember, fermentation is an alternative pathway. What fermentation does is it uses that NADH and produces NAD+. So it actually is using energy, isn't it? NADH is energy, and fermentation uses it. It doesn't actually produce energy. But somebody said recycle. Why is it important to recycle NAD+? Spot on.
But you use it in converting glucose to pyruvate.
Yeah, where do we use it? Well, go back to, let's see, this picture right here. Ouch. I'm just getting attacked by everything today. Garbage cans, a door. It's a dangerous world. Where is NAD+ in this? And where are we in glycolysis? It's not the beginning. Well, it's not the beginning. It's in the beginning of energy payoff, isn't it? NAD+ is right there. Can this enzyme function without it? What is NAD+? What's the fancy word? Oh, gotcha. No. You don't need to know that word. It's nicotinamide adenine dinucleotide. You don't even know that. See, I do know a couple of things. Oh, I'm kidding. What is it called? What is it called? What is NAD+? Other than a substrate here. Yes, ma'am. Yes. She said, I couldn't hear you. Yes. Do you hear? Coenzyme or cofactor. They're kind of interchangeable. Cofactor can be anything. A coenzyme has carbon in it. Okay, so some cofactors are just like an ion. But that, I won't ask you the difference between cofactor and coenzyme. Just say cofactor. But coenzyme is correct as well. What is the definition of a cofactor? Beautiful. Say it louder.
Required for proper function of enzyme.
Required for the proper functioning of the enzyme. NAD+ levels in the cell are pretty low to begin with. So if we're running out of NAD+ because we're making a lot of NADH, what happens to this enzyme? Stops. Well, if we stop here in glycolysis, have we made any money? No, we've only invested money. We didn't get to the payoff, folks. So this is bad news if you shut down glycolysis halfway through, isn't it? Yes, because you didn't get any money from it. You get 0 ATP if you stop here. So that's the whole point, is that when your muscles are starved for oxygen and you can't make ATP through the rest of aerobic respiration, then you run fermentation. Not because fermentation gives you ATP, because it doesn't. But what does it give you? The NAD+. And by the way, fermentation also occurs in the cytoplasm. So where is that with respect to the enzymes in glycolysis? In the same place. So this is in the cytoplasm as well. So this NAD+ right here, this allows glycolysis to continue. And if glycolysis can continue, then we can get to the energy payoff phase where we get our measly little 2 ATP, which is still greater than 0, isn't it? If 0 ATP is bad, 2 ATP is the least function. That's per glucose. So if you burn enough glucoses, you can get 2 ATP for each one. Yeah. Does that make a little bit of sense? So fermentation is not part of glycolysis, and it's not part of aerobic respiration. It's an alternative fork in the road, and it's kind of like a, what do you want to say, a short-term alternative. We're trying to survive until we get enough oxygen, kind of a pathway, isn't it? Because you really don't want to burn, and you don't want to do fermentation too long, because what starts building up again is lactic acid. Well, what does your coaches say you need to do after a big workout? Stretch, rest, drink water. Because what your body is doing, your cells will actually dump the lactic acid back out in your bloodstream. That will go to your liver, right? Your liver is your detox center. Interestingly enough, it'll convert it back into glucose. But that takes rest and energy, ATP. So that's why you're supposed to rest after a big workout.
So like people that struggle with like excessive exercise is it just like you see extra like fermentation in there? And like
Oh, so you're asking about two different things. So like starving yourself and working out hard. Yeah, you can get some fermentation, but really it's oxygen levels that it's dependent on. Probably what your body will start doing is using fats and proteins, which can affect your body in other ways. So you lose a lot of weight, right, if you're not eating sugars. That's those keto diets as well.
Can your body convert fat into something that looks like glucose, or this to like?
Oh yeah. Yeah. And we I have a lecture on alternative pathways, other energy pathways. There's no, I can't remember what it's called. Beta oxidation. And you'll learn this in a few days. Beta oxidation is where you take lipids and cut it into pyruvates. And so pyruvate then will feed into the mitochondria. And, well, not pyruvate, acetyl-CoA. This is Wednesday's lecture. We're going to see it feeds into the Krebs cycle. So yes, you can use lipids. You can use nucleic acids. You can use amino acids. They feed into the citric acid cycle as well. Those all can be used. We've only started with glucose. So you have to realize there's lots of different options to feed into there. Yeah.
So being a distance runner on the track team, I'm just curious, like when I'm like training, am I training my muscles to need less oxygen, or am I like strengthening them to take less? How does that work exactly?
Oh, that's a good question. In my understanding, you're training your body to be more efficient in using the oxygen, but I'm not a medical doctor, so I could be wrong with that. So I would talk to your coach. They probably have more experience in physiology than I do. But that's just my understanding.
Will we have to memorize the drug schedule?
Oh, part of it. Krebs is on Wednesday. We haven't talked about it yet. So we're getting ahead of ourselves. Okay, so if you're asked, why do cells do fermentation, what are you going to say? Just say it. Everybody together, say it. Somebody said about recycling. That's a good way to think about it. What does it do for glycolysis? First of all, does fermentation make ATP?
No.
Does it make NADH? Careful. No, it uses NADH. So it doesn't make energy that way. Well, it doesn't create energy, right? But what does it do? It recycles. Oh, come on. Recycles NAD plus to allow. I thought it'd be so fun if we said it like a chorus. Recycles NAD plus to allow glycolysis to continue. And at least if glycolysis can continue, you can get a little bit of ATP. But the problem is this is a short-term strategy because in animals, lactic acid can start to build up and affect the cells. In yeast, the alcohol starts to build up and kill the cells, basically. So this is only a short-term kind of strategy. There are babies that are born that have mutations in these transport proteins and they're very, very sick because all they can do is ferment. So that's not a strategy for long-term survival. Okay?
So how I understood it, cells that have oxygen can go through fermentation to recycle the NADH to NAD plus. But can it also have oxygen to go through the regular glycolysis cycle and go through fermentation at the same time?
No, because, well, there's no requirement for oxygen in glycolysis, is there? First of all,
The second half of it.
there's no oxygen requirement here. So if there is oxygen, then you wouldn't want to go through fermentation. So typically, you're not going to be doing fermentation and aerobic respiration at the same time. So they're two separate pathways. If you want to think about the pathway homework, you either take one path or the other, depending on what enzymes are turned on. And in this case, this pathway is turned off here. This pathway is turned on by what? Increasing levels of? Yeah, you look like the morning class, like the bus just hit you. It's tough, folks, it's tough. So if we shut off one side of the path, then pyruvate levels rise in the cytoplasm. That's the trigger for fermentation. Really. It's not really oxygen levels, but it's how much pyruvate do you have. And if it's being transported into the mitochondria or not.
So is there some sort of connection between there and this enzyme that allows pyruvate to enter in for respiration to say that we need more NAD plus, so it goes through fermentation to make it?
Well, the connection is that the connection is oxygen, because this enzyme is not sensitive to NAD plus levels, but it's sensitive to oxygen. So if oxygen levels drop, that means we're not getting enough NAD plus, then fermentation is triggered. And it's because pyruvate starts to build up. It's not being transported into the mitochondria. You're going to have to draw it out, look at the steps, think about it. Wednesday, we're going to go down this route here. We're going to see we get a lot more ATP going the aerobic respiration route than going the fermentation route. So for most organisms, that's the preferable route, okay? Okay, practice, 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 practice.